Hello and welcome to the OMGenomics show. I'm Maria Nattestad, and today I'm having a guest on the show for the very first time. This is Robert Aboukhalil. Thank you so much for being with us today.
Thank you for having me. I'm a big fan of the show.
Can you tell us a little bit about your background to start us off?
Sure. So I did my bachelor's in computer engineering at McGill. And after that, I did a PhD in bioinformatics at Cold Spring Harbor Laboratory.
I'm very familiar with that place.
Yes.
The bioinformatics, yes. That's awesome. So what are you doing right now? And what are you doing
right now? I'm a bioinformatics software engineer. So my work focuses on building cloud applications to enable biologists to run complex analyses in an automated fashion and get visualization tools to help them explore their data.
Cool. So what did you do during your PhD? And do you remember maybe the title of your thesis?
Oh boy. It was something like elucidating cancer evolution via single cell sequencing.
Oh, that sounds really impressive.
Yeah.
Yeah. It always sounds a little pretentious when we do our thesis titles,
Exactly.
doesn't it? Yeah.
I
like that. So did that prepare you well for a bioinformatics software engineering position, you feel like?
Yeah. So interestingly enough, into my PhD, I started building web applications to package some of the algorithms and the software tools I had built into a web app to disseminate them to the general biological audience.
That's awesome. So did you find that had an impact on how many people were using your tools?
Oh, absolutely. So I found that, for example, when we would go to a conference and present our work, people would often come up after and say, I've had this data lying around for months now, waiting for our collaborators to finally have some time to analyze it. And now I can actually look at the data myself, and oftentimes they'll say that this is a very empowering tool to have.
So they actually use the word empowering.
Yeah.
How does it feel to empower other scientists with your work? It sounds pretty powerful.
It is. I think it's the best feeling I've had during grad school, which is partly why I decided to continue down that route of building usable software for bioinformatics.
Right. I really like that because I had a very similar experience, partly because I knew you and, we have obviously talked a lot during our PhDs and everything. So that, your work was inspiring to me in some way, that it also helped me to get started with making web applications and so on. And then, of course, I got really into visualization tools, and that's now what I'm doing with the OMGenomics company, is making software for biologists. So thank you for that.
Oh, it's not a problem. I mean, you're now in a league of your own when it comes to visualization. So.
That's so nice of you, but I feel like we've also kind of diverged, where I've gone more into the visualization part of it, because that's where, in my own research, that was the biggest gap for me, was understanding the data kind of to its core, understanding the variation, the copy number, and how they come together. And that was kind of where I felt that my research wasn't reaching its full potential, that I couldn't quite understand everything that was going on in the cell line we were sequencing. And so I started to go into that a little more. But I see that you've gotten more into cloud computing now, especially with your jobs in industry.
Yeah, so my focus has kind of shifted more towards how do you build scalable cloud infrastructure to optimize bioinformatics analysis.
Right, that's really cool. Because I feel like a lot of people, just to say a few words about cloud maybe, since you have so much experience with it, there's probably a lot of people who would treat the cloud exactly like they would treat their institution's cluster. Keep it on all the time and just go on there and run things when they need to, but it'll always be on. So what would you say to that?
Yeah, so in my experience, that's the wrong approach to take. I think you have to think of the cloud as an on-demand service provider. So the best way and the cheapest way to use the cloud is for on-demand kinds of analysis. So if you here and there need to launch an analysis, and once it's done, you just stop using the cloud.
Shut it down.
Right, and
Yeah.
that really takes advantage of the reason why the cloud is so powerful, is because you can scale to so much greater level than you can if you do this in-house. And you can scale in very short amount of time. And then you don't have to pay for maintenance, you don't have to pay for cooling.
Right.
And so
you don't have to hire an entire IT department just to manage it.
Right.
Yeah, so when we say the cloud here, we mean Amazon's AWS, Amazon Web Services, I believe it stands for, and the Google Cloud. So this would be cloud providers. There's also Microsoft Azure. I don't know if that's how you pronounce it. But
I actually have no idea. Azure.
I don't
Tell
know.
us in the comments.
How the hell do you pronounce Azure? All right, so what we wanted to talk about today was a little bit about how whether biologists should use Excel to do some of their data analysis, and what are some of the advantages and pitfalls of that. When do you switch to doing something else? So what would you say about whether biologists should use Excel in their research?
It's funny because when I was preparing for this, I thought back to a year ago. I think if we had done this interview a year ago, I would have said you should never use Excel in biology.
Right.
I don't feel that way anymore.
Okay.
In fact, it's slightly the opposite. I feel like, Excel has its drawbacks, and we can talk about that, but I feel like for the most part, Excel is a very powerful tool, and telling people you should just use R or Python instead is looking over the fact that learning R and Python is not an overnight thing. And so I don't...
a learning curve there.
Right.
And they're just kind of jumping over that, like, we assume that you already know how to use R.
Exactly.
So you shouldn't use Excel anymore. Yeah. So a year ago, what was the thinking that you had? Because I see some of that in the community. There's a bit of a stigma where some bioinformaticians will say that if you're doing all of your data analysis in Excel, then it's not really bioinformatics, or that you should never use Excel. So where do you think that comes from?
Right, so I think this no true Scotsman argument kind of comes from the idea that people who say that already know R and Python.
Okay.
And so to them, not using Excel is super easy.
So what do you mean by no true Scotsman? Because I'm not that familiar with that phrase.
So it's essentially an argument style where if you say, this is not true bioinformatics because you don't do this.
So basically, you're not a true bioinformatician if you still use Excel.
Right. And so similar arguments have been used in other fields, like you're not a true developer if you don't use this or that tool, which is obviously nonsensical.
Right. We should always be using the tools that make sense for our own research. And, we're always just trying to accomplish something. So being pragmatic, I think, is always the best idea. So there are some pitfalls with Excel still, though,
yes.
right? So I want to talk about one of those, but first, at what point do you think that Excel is good enough, and when might you need to switch over into something like R in order to continue your analysis?
Right, so I think Excel is perfectly fine if you're dealing with data that doesn't crash Excel.
Okay.
So especially if you're collecting data, or you just received some, let's say, an RNA-seq matrix of gene expression, and you just want to plot a few quick things, I think that's fine. And
just initial data exploration is
right.
good. Data collecting, I definitely agree with. People in the wet lab who are maybe doing a time trial and they're just entering data in Excel every time they take a measurement. Of course, you don't need to jump over to R in order to do that. You just need to note your data down in some format that makes sense. And then you can export it from Excel into R actually very easily.
Right.
And so that's a really good point. So at what point do you feel like you do have to switch over into R then? Or Python or something else?
Right.
Yeah.
So I feel like if the data gets too large, or if you're doing some complex analysis that Excel just doesn't support. So I don't know, if you're doing some very complex statistical analysis, obviously you shouldn't be doing it in Excel. But quite honestly, I think probably more than 80% of what biologists need is already there.
Until you start getting into NGS pipelines and
Right.
so on, because that's
Exactly.
the whole other beast.
Right.
Yeah. So there's a problem that I wanted to talk about with Excel, which is that some gene names like OCT4 get converted to dates like October 4th, and DEC1 gets converted to December 1. And since this doesn't happen to all of your genes, you might sometimes miss it because it's in the middle of a list that, human genome, you have 20,000 to 100,000 genes depending on how you look at it. So they could be very well hidden in your data.
Yeah.
So what are some of the problems with that? And do you have any ideas for how to solve it?
Yeah, so one of the biggest problems is that a lot of your gene names now get converted to dates, and as soon as you click the save button, they're overwritten. And so if you do any kind of downstream analysis, you've lost OCT4, you've lost DEC1. And so these genes are not going to show up because probably downstream analysis is not going to recognize them if they use any kind of database.
Right, so if they're matching against a database from the UCSC genome browser or something like that, then the gene names will no longer match.
Right.
And you'll completely lose whether OCT4 was differentially expressed.
Yes.
Which could be very important.
Indeed.
Right, so how do you deal with that issue?
Right, so it's funny because if you look at the Excel manual, they'll say that there's no way to completely turn off this feature.
Feature.
Yes.
It's useful, it's not a bug.
Exactly. Yeah. But there are some workarounds. So one of the workarounds, which is slightly tedious, is to edit every single gene in your text file and add a single quote before it. And so that way Excel is going to leave it alone and not convert it.
So it's not quotes on both sides, it's just a single quote, like an apostrophe, right before.
Unclear to me why that is.
That's odd. Yeah.
But yeah, this is one of the things they suggest on their help page. Now there's a slightly better way of doing this that doesn't involve you editing every single line.
Good.
And it's never double click on a CSV or TSV file and have it open in Excel, because then the conversion is done already. Instead, you want to go to File, Import, and then it'll give you a wizard. And then when you get to the step where it shows you all the columns in your data, you need to select the column that contains genes and select the text instead of general. And so a general column will get converted to dates if it looks like a date, but a text will just stay as is.
And it doesn't have to be that the whole column looks like dates in order for it to do it. It'll do it for each individual cell.
Yes.
Which is why it's so dangerous, because it doesn't say, oh, this is not a date for all of your other genes. Right?
Yeah.
Yeah, that's pretty bad. So you can do it through adding a single quote before every gene name.
Yes.
I recommend Sublime Text for that if you're doing that kind of analysis, because you can highlight all of your columns and add the same character to all the columns at once, which is really cool if you hold down, I think, Option. So that's one way to do it. I like your way about doing it in the wizard in Excel, but I think you mentioned to me that you also have a little application now that helps you do that. Yeah, so it's called OCT4th?
Yes. And so
I like the name.
yeah, it's kind of highlights the issue that it has.
Right. So what does the app do?
So essentially, it allows you to select a bunch of CSV and TSV files, and it will convert them into Excel files without doing any kind of funky conversion into dates.
Oh, cool. So that takes care of it. Okay, where can we find that application? I can put up a link here.
Yeah, so you can find it on Gumroad.
Okay.
And yeah, we can put a link to the app, and it's totally free, so you can get it today.
Oh, awesome. So we'll put up a link maybe like right around here, and you guys can go check that out. So before we wrap up here, is there anything else you want to talk about? And then we can give some links to your websites and so on.
No, I think that covers it. I think that in general, telling biologists don't use Excel, just use R or Python is bad advice. We should be telling them, look, Excel is good for a lot of things, just be aware of these issues and here's how you fix them.
That's very true. and understanding maybe what kind of statistical tests Excel can't do.
Yeah.
Google is your friend. You just
Exactly.
you go and you search, can I do this in Excel?
Yeah.
And when you hit upon something that you can't do, instead of saying, okay, then what? Maybe I won't do that for my research. Maybe this project doesn't need a t-test. I don't know.
Right.
Then you should probably go and find another way to do that. And that's when, for people who've been using Excel and haven't done anything else, I would recommend R as your second step for when Excel falls short. But see whether you can use Excel the whole way through, and when you run into the problem, that's when you switch.
Absolutely.
I don't agree with people who say absolutes like you're not a true bioinformatician, or you should never use Excel for doing biology research. It just doesn't make any sense. We always have to be pragmatic.
Yep.
Yeah, that's one of my moral codes, is pragmatism.
Excellent.
All right, awesome. So where can people find out more about you and get in touch with you on social media or something?
Oh, so you can find me on my website, robertaboukhalil.com, or I'm also on Twitter @RobAboukhalil.
Awesome. All right, so thank you so much for being on the show, and we'll wrap up here. I want to say to all of you viewers that you can sign up for email updates from OMGenomics.com at omgenomics.com/subscribe. And that way I'll send you weekly updates when new videos like this one come out. I hope to do some more interviews in the future because I think this was really good. And you can also subscribe on YouTube, and that also helps other people find these videos, so we can spread this kind of knowledge around the community because I think it's important. So thank you so much for watching, and I'll see you next time on the OMGenomics show.